Our research is really looking at how changes in cell signaling can lead to disease. In the heart, we can see changes in calcium leading to changes in rhythm. And these changes in rhythm is something we need to measure. So the reason we turned to the CAHA system is because we can do long-term recordings of ECGs. Similar changes of calcium in the brain can lead to altered function, specifically seizures and dementia. Again, we turn to CAHA because we can do really long-term recordings in these animals to see how brain function changes over time. Most of the time with these systems, you can record for a short period in blocks. And that can be really useful in certain types of disease where you're giving a stimulus to trigger a result. Whereas if you're looking at more chronic diseases such as heart failure and arrhythmias, or neurodegenerative disease, which gets progressively worse over time, you can't take these snapshots. And a big limitation of previous research is you can say, give an animal a heart attack to change its cardiac function. You then record for an hour or two, and then your data is done. You then do another experiment where you can record for an hour or two, but 2 days later. Or you do it a week later or a month later. But you're missing everything in between. What we can now do is record continuously through that time to see how the function of the heart is changing over time. This is even more important when we start thinking about neuronal function. Alzheimer's disease, for example, is progressing over weeks and months. Currently, all of the research really looking at brainwave activity, especially in rodent models, again looks at these snapshots. Or it has to use a triggered response where you inject the animal with something to trigger a seizure, and then you can measure what happens. What we can do is measure these spontaneous seizures over massive lengths of time. We can see how they change over time, how they might build up, and how they change the function of the animal. Everybody knows dementia is really common, Alzheimer's in particular, but also people will be fairly aware that the treatment options are really limited. An issue becomes of not only when do you treat the disease, but also when can you diagnose the disease, because of course you can't start a treatment until you know it's occurring. One of the hopes is by measuring EEGs, we might get some early indicators that these animals are going to progress into a more severe form of the disease. This, in theory, could be translated to patients, whereby using more detailed EEG monitoring, we might be able to identify those patients that are most likely to progress the fastest into disease. If we combine this with our work looking at the treatment of Alzheimer's disease, these are the patients we could potentially put on our drug study earlier in the process to try and stop that deficit in memory and the other complications that come with the disease. This is a major concern for New Zealand. We're getting older. Thankfully, for many other diseases, we're not getting those as common. Therefore, as our population gets older and older, we're going to need more treatments for these neurodegenerative diseases.